Dear student, today we will discuss polysaccharides. The polysaccharides are the polymer of monosaccharide in which the monosaccharides are joined together to form long chain called polysaccharides. And these are associated by glycosidic linkage. During this, the molecules of water are lost, and this is how the polysaccharides are formed. So they are insoluble in water. Now, on the basis of composition, the polysaccharides are divided into two categories: homo and heteropolysaccharides. Homopolysaccharides are formed by the polymerization of one type of monosaccharide such as glycogen, starch, cellulose, whereas heteropolysaccharides are formed by polymerization of more than one type of monosaccharide such as peptidoglycan, chitin, etc. Now, based on function, they are divided into three categories: storage polysaccharide, structural polysaccharide, and mucopolysaccharide. So, storage usually they are all homopolysaccharide, and they are stored as reserve food material in organisms such as starch in plant, glycogen in animals. Structural polysaccharide, they are also homopolysaccharides and also heteropolysaccharide, and they take part in the structural framework such as cellulose, which is present in the plant cell wall, chitin, which is present in the exoskeleton. Then mucopolysaccharide, they are all heteropolysaccharide, and they consist of repeated disaccharide unit. They are mucilaginous substance such as peptidoglycan, heparin, agar-agar, pectin, hyaluronic acid, chondroitin sulfate, and algae. Okay. Now, I will be discussing some of them. First is the starch. You know that is starch is the polysaccharide which is made up of glucose unit joined by alpha 1,4 glycosidic linkage. So the starch is a branch structure. At certain point the branching also appear, and wherever the branching occurs, it is by forming the alpha 1,6 linkage. Okay. So when you do the hydrolysis of starch, then one is the branch and one is the unbranched portion that is formed during the hydrolysis. Okay, so the unbranched portion is called amylose, which consists of glucose, in which the glucose are binded by alpha 1,4 linkage, whereas amylopectin is the branch chain of glucose, which consists of alpha 1,6 linkage. If you do the chemical analysis with iodine, you know that starch produces blue color with iodine. So this amylase will form blue color with iodine, and amylopectin will form red color with iodine test. Okay. The next is glycogen. Its structure is similar to starch. It is also having branching, but these branching are not much as they are present in the starch. So the glycogen carry alpha 1,4 glycosidic linkage and alpha 1,6 glycosidic linkage, and it is present in the it is present as a storage of glucose in liver and skeletal muscles of animal. Cellulose, which is composed of beta D glucose. So it is a polymer of beta D glucose where the glucose units are joined by beta 1,4 linkage, and this is not branch but it is a straight chain, and it can form microfibril with high tensile strength. They are present in the plant cell wall. In human, it acts as a roughage and it is not digested in GI tract, whereas in ruminant, it is digested since they have four stomachs. So one of the stomach is rumen, in which the bacteria ruminococcus is present, and these bacteria can synthesize the enzyme cellulose that act on beta 1,4 linkage and digest. Since the human cannot synthesize the enzyme cellulose, so cellulose is not digested in human. Now, if you look at the chitin, it is the linear polymer of N-acetyl glucosamine. Now, this is the derivative of glucose. You can say at second position, this special group is added where the H is replaced by NHUCH3. So this is, if repeating units are present, we say this is N-acyl glucosamine. Okay. So chitin consists of this, and we have already seen it is present in the cell wall of fungi, it is present in the exoskeleton of many invertebrates. Peptidoglycan, it is hetero polysaccharide. We have seen it is present in the cell wall of bacteria, and it consists of repeated unit of disaccharide. So out of that, one is N-acetyl glucosamine and another one is N-acetyl muramic acid. So N-acetyl glucosamine, that is NAG, and N-acetyl muramic acid NAM. So NAG, NAM, just consider this as a one unit. So this keep repeating NAG, NAM, NAG, NAM, NAG, NAM. Okay, and it occurs in the cell wall of bacteria. Hyaluronic acid occurs in the matrix of connective tissue and act as lubricant in synovial fluid of bone joint. It forms cerebrospinal fluid of brain and spinal cord and vitreous humor of the eye. Heparin, it is a sulfated mucopolysaccharide which is produced by liver and act as a anticoagulant. Keratin sulfate, polymer of N-acetyl glucosamine, galactose and sulfate present in eye, skin, cartilage, bone and connective tissue. It gives strength and flexibility. Then agar-agar, it occurs in algae. We have seen in red algae. It is one of the product, and it is the polymer of D-galactose molecules. It is used as laxative and solidifying, used to form the solidifying culture medium for the microorganisms. Pectin occur in the middle lamella of plant, and it is polymer of galacturonic acid. Again, these are all derivative of the glucose, and galactose, arabinose and methylated galacturonic acid. So these are certain polysaccharides. You must know what is their significance. So this is all in polysaccharides.